Okay, so yeah, I'll be speaking about GeoTA tool, the tool that is used to analyze spatially resolved transcriptomics, the method that was named the method of the year by Nature. Well, it was a great breakthrough when single cell was developed. Compared to bulk DNA, it allowed us to specifically study separate cells. But when we analyze data using single cell, we end up with something like this, just cells in an artificial dimensions. In this case, it's UMAP, but we never really know where exactly in our tissue these cells are located. And with spatial transcriptomics, finally, we have this opportunity to map the cells to our tissue. Like here, this example, this is the kidney, and this is normal kidney, injured kidney, and another injured kidney. And we can see that, for example, our cortex is comprised of a mixture of different cells that are located here, the glomerular or the collecting ducts. And our other cells, for example, proximal tubule cells, this third segment is located here. It's pretty much all the outer medulla is comprised only of this type of cell. But other than organs, we also can look at, for example, biofilm populations. This is the clustering of different functions that cells, individual cells in the colony do. And using spatial transcriptomics, it was possible to map them to the specific positions in the colony, which allowed to identify some hotspots for functions. For example, here we can see that this hotspot does the denitrification. Here we can see it's hotspot for high oxidative stress and so on. Of course, it's not the only application of spatial transcriptomics. It's used a lot in medicine, for example, in studies of cancer, because tumor is very heterogeneous and there are different clones. Some parts are evolving faster, and of course, we can see some differences in expression. Also, it's very useful to identify the invasive margin of the tumor and actually how the cells in this cancer interact with normal cells. And it was possible to build a bunch of expression atlases for healthy organs or the ones in disease. Also in molecular biology, it allowed to localize the individual molecules and, of course, study their expression inside the cell, for example, in cytosol or in nucleus. It also helped to understand better the chromatin organization and cell development. So there are different experimental platforms to study spatial single cell. We can separate them into microscopy-based methods like SECFISH, OSMFISH, or MERFISH, all the hybridization-based methods, and sequencing-based methods, for example, division. The difference is that for microscopy-based methods, since it's based on hybridization, we have a single cell or even single molecule resolution. And thus we can identify their locations if it's in cytosol or in nucleus or somewhere else. But we're restricted to only known genes, and we cannot study any novel ones. And we also can't analyze too many genes at the same time because we're restricted in number of colors, or they can just basically overlap and we won't see exactly where this molecule is. Sequencing-based methods are easier and cheaper, and they don't really need a lot of pictures made on microscope to study the locations. And we also can study novel genes that we don't know the sequence of, but it's not always a single cell resolution. And we will be focusing on vision, that is exactly not single cell resolution, but basically how vision works. We have a plate like this, and the capture area that is comprised of small spots And each spot already has the barcodes, the spatial barcodes that will tell us where exactly this position is located. So we place our tissue above the capture area, and we treat it so the mRNA will ligate to our barcodes. And then we just proceed to sequence, and we get a sequence like this. So after sequencing, we end up with our matrix where we have gene counts per cell and cell coordinates. And of course, we can get some pictures to work with as well. So with the data I just showed, we can start our pipeline, and spatial transcriptomics is actually very similar at the beginning. It's very similar to single cell analysis. It also has the pre-processing steps, the dimension reduction, different clustering algorithms, and different algorithms for cluster-specific marker gene identification, cluster annotation. But Giotto also allows us to check, to play with clusters, to separate them or to merge them based on their similarity, and run enrichment. And this module, the spatial analysis, is what is really different and interesting for us. It starts with building spatial network, and then we can find spatially variable genes that have some spatial pattern and study co-expression and cell neighborhood. So I will focus only on this part. So we already did all the pre-processing and single cell analysis and end up with our artificial dimensions. In this case, it's UMAP. And we color them with latent clustering. So in this case, we have 11 clusters. This is kidney sample. And what is different from single cell at this moment is that we already can map our cells on our tissue. And we see here that, for example, clusters 6 and 7 comprise the inner medulla, and classes 2 and 9 comprise the outer medulla, and the rest of the classes comprise the cortex, and it's really a mixture of cells. What we can see here is, for example, we can plot how many genes are expressed by which part of our tissue, and we see that medulla is expressing a lot more transcript than the cortex. So the first step in spatial analysis is to plot a spatial grid or network that will allow us to calculate the relationships between these parts. In case of grid, this is the expression will be summarized inside the grid. And in case of network, it will remain as a single cell or single spot resolution. And this allows us to run spatial gene search. And I wanted to show you an example of two genes, NAPSA and GPX3, which are both marker genes of proximal tubules. But you can see on these plots that they actually have completely different spatial expression patterns, right? We can see GPX3 expressed in the cortex and not expressed in the medulla, and NAPSA is expressed in outer medulla. So comparing it to already known cell types in the kidney, we can see that actually GPX3 is a marker gene for proximal tubules, but segments 1 and 2, which are located in the cortex. And NAPSA is more correlating with proximal tubules, the segment 3, which is located here in the outer medulla. So spatial analysis can help us understand marker genes better. Also, the next thing is spatial co-expression. So this actually allows us to identify the metagenes, a group of genes that are sharing same patterns of expression. And for example, for this data set, 8 metagenes were identified, and here we can see the correlation between these 8 metagenes. And here are the expression for the metagenes on our tissue. And you can see that they all have different patterns of expression. Some are specific to inner medulla, some are specific to outer medulla, some are specific, well, not quite, because here's a mixture of cells really, but they're specific to the cortex. And we can go from the metagene level to single gene level if we want to. And one more, maybe the most interesting thing, is how the cells interact. And in this case, it shows how really, well, the first step is to find the interaction changed genes. So the genes that actually change when their expression changes when there's interaction with different cells. And based on these genes, we can find cell types that are interacting within this cluster or with other clusters. And we can see, for example, that clusters 4, 5, 8, 1, and 3, they all are here. They aren't really correlating even to each other very well. And for example, cluster 10 and 11, these are small ones. They're located here and here. They have very strong interaction inside the cluster and not so much with other clusters. Well, they interact with each other as well. So a spatial single cell really gives us a lot of opportunities to study pretty much anywhere we can apply single cell. We can try applying spatial single cell, and it will give us more information at where exactly this function is happening, and a lot of information about expression patterns, spatial expression patterns. So that's it on the GOTO. If you have any questions, I think you can type them in the QA section if there is one here. And I will move on to amplicon sequencing, right, Julia?
Yes, thank you very much for this introduction to the spatial.
I think we do have. I think we do have a couple of questions if we can answer them.
Okay, yeah.
Yeah, so the first question is, how do you select between various manifold methods when analyzing complex data, such as spatial single cell?
So I think, yeah.
Yeah, sorry.
You want to answer? Take that, Lisa, sorry. You can take that.
No, I mean, how I choose, I just look at the data, I guess, and see if it makes sense or not.
I would also suggest that you can use, there is no right embedding. So and I think it's an important issue where it's an important thing to remember, that you're not trying, each of those embeddings is going to be somehow right and somehow wrong. It will preserve some of the hidden structure of the data, but it will lose some other hidden structure. So by using many different methods, you can find different, sometimes it's trajectories that you're interested in, sometimes it's clusters that you're interested in. So we always try to recommend using as many methods as you can to look at the data from different sides. So also such complicated data as single cell or spatial single cell, the UMAP is actually most, if you look at all the articles, you will see that most of the people use UMAP or TSNE, but there are many, many, many more embedding methods available, and we recommend to really look through at least a few, because some of them, they're based on different things, and then they also will reveal different underlying structures of the data. So I hope that helps.
Great, thank you both. Another question, will spatial single-cell RNA-Seq make single-cell RNA-Seq obsolete, without imaging data obsolete?
Can you please specify what obsolete is? Sorry.
Yeah, so will single cell RNA-seq without imaging become irrelevant with the introduction of spatial single cell?
Well, I think that there are options when we don't really need spatial imaging or we can't really capture it. Well, looking at the pipeline, yeah, it looks like all the steps we do the same as single cell, so we can replace all the single cell projects with spatial single cell. And I think that's a good idea, but still, I guess there are options when we can't really use spatial single cell, so we can continue working with only just regular single cell.
Also, if I could add to that, if you are using Visium, then as Lisa has mentioned, you are not actually capturing each individual cell. You are capturing a small well that has a few cells in it, right, Lisa? So probably, at least for now, single cell is still the resolution of the single cell RNA-seq is probably better than at least this Visium. And another important thing for science is a matter of resources. Spatial single cell is a very expensive technique. We cannot imagine that all of our studies will become all of a sudden only single cell and only spatial single cell. It will be just too expensive to do science this way. So for a well-identified project, we hear prices around $100,000, $200,000. So not every lab can actually afford this.
Actually, one more thing, Juliet made a good point that Visium is not single cell resolution. There are studies when just regular single cell and spatial single cell are combined together. So you get the spatial information, but at the same time, you can kind of get the single cell resolution. So yeah, I think single cell won't go anywhere. We'll use it.
And if to add a little bit to that, a lot of people ask us if now we have this technology that allows us to do single cell, will bulk RNA-seq be obsolete? And the answer is probably related, probably not. There are more and more computational ways of combining single cell data, which is very sparse. So you have a lot of genes that appear to be not expressed at all, but in reality, they are expressed. So the data is very sparse, and you combine it with your bulk RNA, which is much more stable, but has this average cell that is never actually exist. And you do something like deconvolution and use other computational techniques to try to connect those two together. So I think it's the same kind of question.
Great, thank you so much. All right. Now I
just
can
a minute.
share.
I think now we will have Chime. And Lisa,
yes, I was going to show the recorded presentation. Is that correct?
No.
It's a different one. Maybe
No
yours is the next one. Sorry. Can you see my screen?
Yeah.
Okay, yeah. I'll speak about our platform section for Chime. Chime is a tool that analyzes metagenomics data, the culture-independent microbial communities. What we basically learn about them is composition and function of these communities. And it can be applied in a bunch of areas like agriculture, where you can study soil properties or any bacteria-driven processes. In healthcare, where we can study microbiomes of different organs, or ecology, where biosensors are developed using metagenomics, and biodegradation of plastic is also a bacteria-driven process. And of course, biotechnology, the nutrition produced or pharmaceuticals or biofuel, everything is done thanks to bacteria. So basically, when we sample our data and we extract DNA and then we want to amplify it, we can go two ways. We can either go whole genome sequencing way or target sequencing way. And in case we want only to sequence the target, 16S ribosomal RNA is the region that mostly is targeted in bacteria because it has both conserved and hypervariable regions. Actually, there are 9, just I didn't have a picture good enough for to include it. But these hypervariable regions help us differentiate between different species. So 16S or target sequencing or amplicon sequencing, it's cheap compared to whole genome sequencing. It's pretty standard. We already know all the primers, so it's easier to conduct. But it's restricted to previously known and annotated taxa, and sometimes it's not enough to differentiate between the samples. Also, we only get the compositional information. We don't know any function that are done here. Whole genome sequencing allows us to study novel organisms, and we can assemble the reads in, and we can run functional genomics and understand what function this organism does. It's also more information to differentiate between the taxa, but there are a bunch of problems to whole genome sequencing of bacteria because it's expensive, it's hard to understand what is contamination, what is not, and so on. So on the platform, we present 3 modules, pre-processing that consists of filtering and denoising algorithms. Then we go on to taxonomy with silver classification, filtering our classified sequences, and conducting phylogenetic trees. And then we can move on to metrics to calculate for alpha and beta diversity. So at the very beginning, we start with the quality control, and it's very important not to include regions of low quality because they will interfere with all the downstream analysis with our abundances, with our phylogenetic tree and taxonomy and everything. So you have to understand where you want to trim your reads like this, so you will not include low quality nucleotides in your downstream analysis. So after filtering the replication, we come to denoising or clustering techniques. So the idea is that when we first sample our data, we have one sequence per species or something like that. But when we run amplification, there are bound to be some errors, and we get small fractions of sequences that are slightly different from our original one. And when we sequence, there are even more errors and more unique sequences. And so we end up with a plethora of unique sequences, and clustering algorithms would look for the most abundant sequence and assign it as a center and cluster everything that is 97% similar in one cluster. Well, there are a few problems with this approach. And first is that not all the species are 97% similar. Some are a lot more similar. And researchers came up with another approach, denoising, which was taking into account different models of errors and sequencing quality calls to try and correct the errors, the mistakes, and end up with haplotypes. But these haplotypes are 100% similar. So after denoising or clustering, for paired end reads, there is also option to merge, well, not an option, it's an obligatory step to merge the reads. Since even for targeted sequencing, we can pick different size, different regions, and the size of region may vary, and sometimes the reads are separated like this. Sometimes they overlap, and for example, DADA2 treats them a bit differently. In case they overlap, they only take the part that overlaps. In case they overhang, it will take one of the reads. And if they're separated, it will concatenate the reads together. So when we sampled our data, extracted DNA, amplified it, and did denoising, we end up with a table like this, where we have counts of each unique representative sequence per sample. And here we can ask ourselves three main questions. First is, how similar or different the things are in one sample? And that will be alpha diversity. Second question is, how similar or different are the samples between each other? And that will be beta diversity. And the third is, what specifically leads to differences between the samples? And that will be taxonomic profiling. We'll start with the third one. So for taxonomy analysis, it is strongly advised to use the same primers that you used for your data. So the same region will be extracted from the database. Here we use Silva database that is being updated and curated. And you can see the mostly used primers here and primer sequences. So you just take the sequence, add it to the platform, forward and reverse primers, and they will extract the exact regions from the database and run classification. So it will look for if this sequence is in the Silva database, and it will assign taxonomic unit to the sequences. And you will end up with a bar plot showing what bacteria are there in your samples. If you want some of them removed, you can exclude them. Just type the correct name of the taxa, or if maybe you want to include just several samples, you can do the same. And you can also group the samples based on some categorical columns in your metadata. For example, I was interested in body sites to see if samples from gut were any different from palms and tongue. So after taxonomy analysis, we go on to phylogenetic tree. So this is this will show how similar our representative sequences are, and it starts from multiple sequence alignment, and then there are a few algorithms are available to build the tree, fast tree, and IQ tree. This is mainly used for core metrics, but you can also extract the tree and visualize it in, for example, Fig tree or any other tool that helps visualize phylogenetic tree files. So after that, we have, well, looking at this table, the artificial, I just came up with it to show. Looking at this table, we see that total counts for each samples are different. And in case we can compare sample 4 and sample 1, they're pretty close in total counts. We can't really compare sample 3 with others, right? So because they have different sampling depth, and we can't really run any reliable statistics on something like this. So one of the ways out is to subsample the frequencies so that the total count will be the same for all the samples, like here. So we subsample all the table and change the table so that the total will be the same. This is not ideal because we'll lose information. Sometimes we even lose whole samples if their sampling depth is lower than what would peak. But this is a good way to normalize your samples, so the statistics will be reliable. But how actually should we pick the value? Well, for example, how did I pick 100? We should be looking at our samples and their sampling depth. For example, here, we can see that some samples have sampling depths lower than 1,000. Some have sampling depth a bit less than 2,000. some are here, but the majority are having bigger sampling depth. Also, we can see that the number of features are quite stable starting from here. So it looks like this region looks appropriate to take the sampling depth. But it's also very meaningful to look at your groups and if the samples in your groups of interest are still there. For example, if I take sampling depth of 2000, my right palm sample group will consist of only 2 or 3 samples. But if I take sampling depth 1000, my right palm group will be okay. I can still work with it. The only thing that is left is core metrics, basically calculating all the metrics for alpha and beta diversity using the sampling depth that we just identified. And it will give us an idea. So the alpha diversity will give us an idea how things are inside the sample, basically telling us how rich and even our samples with different indices, Shannon index, Philo index. And beta diversity will give us an understanding how samples are between each other, plotting PCA based on different distances like Jacquard or Bay-Curtis. And we also can run Permanova test on a column that we are interested in. In this case, it was body side. And I got really good p-value, meaning that this group has very different beta diversity statistics. That's it. Thank you very much for your attention. And yeah, here I just mentioning some studies that were done using amplicon and whole genome sequencing of metagenomics. Thank you.